to let everyone join. Great, people are still joining. Welcome to tonight's webinar, Gene Therapy Clinical Trials for Genetic Cardiomyopathies. This is brought to you by the Genetic Cardiomyopathy Awareness Consortium, and you can always go to our website for more information. You see it there at the bottom, geneticcardiomyopathy.org. This is in conjunction with the DCM Foundation, of course. So let's get rolling. Next slide, please. We have some exciting information to bring you tonight. Again, you are here to see the webinar on gene therapy clinical trials for genetic cardiomyopathies. Thank you for joining. We have a few more people that have just hopped on, so give them a chance to get settled. Everyone's microphone is on mute upon joining so that we can hear all the presenters. Good, so let's get rolling. Next slide. Great, we ask throughout the presentation, if you have any questions, there will be a Q&A portion after the presentation tonight. So please drop your questions into the Q&A feature of Zoom at the bottom of your screen at any time, and we will handle all of the questions at once during the Q&A portion of tonight's webinar. Again, all microphones are on mute, just so that we can hear everyone speaking. Next slide. And behind the scenes with us tonight, I have Justine, as always, helping. Thanks for your help, Justine. So first, let's do a quick poll. Justine, if you could please push the poll forward. We just want to know who's attending tonight's webinar. What best describes you? Are you a cardiomyopathy patient, a family member or friend, medical professional, or industry professional? And we'll give people just a minute to answer and decide what they are. Excellent. Great, let's share the results. We have 41% cardiomyopathy patients, almost 20% family member or friend. Great, we figured we'd have a lot of folks on who are patients or family members. Medical professional, almost 20%, and industry professional, 24%. And this is a very hot topic, so we do have, of course, a lot of interest from the medical community and the industry itself. So we can stop sharing that and let's continue on. Next slide. Again, tonight's presentation is brought to you by the Genetic Cardiomyopathy Awareness Consortium in partnership with Lexeo Therapeutics. We're so thrilled to have Lexeo as one of our sponsors and really a great supporter of everything that we're doing with GCAC. Whenever you hear me say GCAC, that refers to, that's just an acronym for the Genetic Cardiomyopathy Awareness Consortium, which is a mouthful. So next slide, please. So tonight's topics, and our presenter tonight will go into these in more depth. What is a gene therapy clinical trial, and basically how is that different from other kinds of trials? We'll then go into the phases of gene therapy trials here in the US. We'll talk about potential considerations and support from a patient perspective, how to then identify a trial that's right for you, and then a little overview about some of Lexeo's work in this area, specifically for the PKP2 arrhythmogenic cardiomyopathy, and then, of course, our live Q&A. So next slide. So tonight's guest speaker, I'm thrilled to introduce Nilomi Shah from Lexeo Therapeutics. She is the Medical Affairs and Patient Engagement Lead for Lexeo. She has 10 years of experience working in medical affairs, and she's supported development of various gene therapies for the last 5 years. Nilomi received her doctorate of pharmacy from the Ernest Mario School of Pharmacy in 2014, and we're thrilled she could be with us tonight. So next slide. Nilomi, thank you so much. We will hand the controls over to you, and if you want to share in your deck, and then you can take it away.
Perfect, thank you. Just get the ball rolling here.
Excellent.
Can you see the slides okay?
Looks great, and then we'll spotlight you, and I will be right here behind the scenes.
Okay, thanks, Jan, and thanks to the DCM Foundation for inviting me to give this talk on gene therapy clinical trials. So just very quickly, some disclosures. Like JN mentioned, I am an employee of Lexeo Therapeutics, and Lexeo is researching an investigational gene therapy for arrhythmogenic cardiomyopathy due to the PKP2 mutation. So clinical trials and gene therapy clinical trials in particular is a big topic. There are a lot of details that we can go into. So my goal really for the webinar today is to just touch upon some of the important aspects of this topic. And the hope that you have the information available to then ask questions and really understand whether a gene therapy trial may be right for you, your family member, friend, or support system. So during this webinar, I hope to help answer the following, starting from the very beginning. What is a clinical trial? What is the purpose of conducting a clinical trial? What does the typical drug development process look like in relation to the clinical trials? You'll hear me use the word phases sometimes, and we'll dig into that in a little bit. How may gene therapy clinical trials differ from non-gene therapy clinical trials? What are some potential considerations for participation? And then where you can find some information on active clinical trials. I'll also take the opportunity towards the end of the webinar to share a little bit about Lexeo's gene therapy program for arrhythmogenic cardiomyopathy due to the PKP2 mutation and our currently enrolling HEROIC PKP2 trial. So before we dive into the actual webinar concepts, just wanted to take a little bit of time to introduce Lexeo so you know who we are. Lexeo currently is solely researching gene therapy. And as you see here, there's really two main areas of focus for us. So Lexeo was established in 2017. You might consider us a new kid on the block. But as you can see here, we have a lot of activity in a deep cardiac pipeline. Including what I just mentioned, an active clinical trial for arrhythmogenic cardiomyopathy. We also have an active clinical trial for Friedreich's ataxia, which is a neurodegenerative disease, but we are targeting the cardiac aspect of that disease. And we hope to share more information on both of these trials, some preliminary data later this year. I should also mention that we have some programs for desmoplastic cardiomyopathy and hypertrophic cardiomyopathy as well, although those are not in the clinical stage of development. And this is me, just a little bit about me since we'll be spending the next hour together. Like JN said, I have been at Lexeo for about 2.5 years. I'm a pharmacist by training, and in my role in medical affairs, a big component of what I do is working with advocacy groups to educate, inform, and support the communities that we're researching gene therapy in. All right, so let's get started. What is a clinical trial? So before any drug or gene therapy is approved for use in humans, it must undergo a rigorous testing process called a clinical trial. By the FDA's definition, a clinical trial is a research study in which people volunteer to help find answers to specific health questions. So these specific health questions can kind of be put into two main buckets. One is around safety. Does the investigational drug that you are researching cause any harm? And the other is efficacy. Does the investigational treatment work? And the way that we look at that is researchers measure if the treatment is improving the condition and disease and it's meant to do what we think it's going to do. So what's the purpose of a clinical trial? In addition to looking at the safety and effectiveness or efficacy of new treatments, the data that's collected in clinical trials helps to compare that new investigational treatment against things that may exist already for that disease or condition. So other approved medication, in other words. And we're really evaluating whether this new investigational treatment is improving, improving upon what already exists or filling a specific gap. There are instances, and it's typical for the diseases in which we're researching gene therapy that nothing is approved. And so really what you're comparing against is just things like symptom management. Other things that clinical trials help to answer are, how innovative treatments like gene therapies actually work in the human body. So there are studies done in the lab and animal studies that are conducted that give us a good idea of how the gene therapy may work, an educated hypothesis. But a clinical trial is really the first opportunity for us to understand how something like gene therapy can work in the human body. The other thing that clinical trials help to do is collect really important safety information. So things like potential side effects, if any. For a treatment, any treatment to be approved, you must demonstrate that there is more benefit than risk associated with that treatment. And so this is just another way of collecting that information to help answer that question. So what does the typical drug development process and phases of clinical trials look like in the United States? So before we dig into this, one thing that I wanted to mention that's not on the slide is that we don't, and we being the company and others, don't automatically jump into testing in humans right away. There are other steps that happen before this. So studies in the lab, studies in animals, where all this data is collected looking at the investigational drug. That data is then put together and submitted to the regulatory body in that country. So for the US, that'd be the Food and Drug Administration, or the FDA, who carefully reviews that data to make sure that there's no signs of safety concerns and that there is some potential benefit to justify moving into a clinical trial for humans. And so there isn't this automatic jump of just going directly and testing a new treatment or investigational treatment in humans. There are other steps before that to make sure that that it's justified. So clinical trials are typically conducted in what we call separate phases. Each phase is helping to answer more questions and to better understand the treatment. One thing I should mention is that no two clinical trials are the same. Think of them like snowflakes. What you are looking at, the amount of people in a phase of a study, the length of the study is really dependent on the patient population, the disease or the condition that you are researching, and the type of therapy that you are investigating as well. So starting for the first row, phase 1 studies are typically done in a small group of people, and really what you're looking at is safety. Safety is the most important thing. So you're looking to see if there's any side effects that you're seeing associated with this within the phase 1 study with the investigational drug. It may also help answer questions such as how does the body process the drug, how much and how often a drug should be taken, once a day, twice a day, once a week. Obviously, with gene therapy, the hope is a one-time treatment, so that's a little bit of a different situation, but just talking in generals right now. And the length of how long a phase 1 study may last could be anywhere from several months to even several years. Again, really depends on the patient population and what you are investigating in the disease. Phase 2 studies are a little bit larger in size. You are still looking at safety. You may also continue to look at and answer the question around how the body is processing the drug, how much and how often it should be taken. But this is where you start to see signals for efficacy. Is the drug actually doing what we think it's going to do? Is it showing any signs of benefit? And phase 2 studies can also last months to years. And then finally, phase 3 studies, these are the largest. And when I say large, this could be several hundred people, this could be several thousand people. You will continue to look at safety in phase 3 studies. At this point, you should kind of know how the body, the drug is being processed and how often to take it, but you're really digging into how effective the drug is. Does the benefit and the data collected show that there is benefit in this investigational treatment? And does that outweigh any risk? And because phase 3 studies tend to be longer, we're talking several years, up to a decade even, it's an opportunity to look at the long-term side effects and efficacy of an investigational treatment. After a phase 3 trial is completed, all of this data is collected, put together, and submitted in an application to the FDA, who carefully reviews all of the data to see, one, is the investigational drug adding any additional benefit to what already exists, right? Comparing it to what may already be on the market and is approved for that specific disease or condition, and does the benefit outweigh the risk? And there's other things that are involved with this, of course, but essentially, if they deem that that is the case, then a drug will be approved or is considered approved for and available for wider use. I won't go into phase 4 studies too much, but essentially this is a way to really look at data in large forms. Now that the medication is approved, it's available for people outside of clinical trials. You can really track the longer term effects of both the safety and the efficacy. I think the most important takeaway here is that safety is the priority in each phase. If a study is not deemed safe at any point, it will not move on to the next phase of development. A lot of trials have groups called institutional review boards, IRBs, or data safety monitoring boards. These are, third-party independent groups that are looking at the safety data in an unbiased way to ensure participant safety and to make sure the trial is being conducted ethically. So we talked a little bit in more general terms about clinical trials. Let's focus on gene therapy and what might make that a unique approach. So unlike traditional medications, we'll call it, which are really managing the symptoms of a disease, gene therapy really aims to target the underlying cause of the disease, many times a mutation in a gene that is causing the disease or the condition. Just like any new treatment, gene therapies also have to be tested in clinical trials. And there is a similar drug development process for gene therapies, but there are also some important differences and considerations that I'll dig into. So in addition to looking at efficacy and safety, another thing that's looked at for gene therapy clinical trials is the gene that you are introducing into the body going to the target cells that it needs to go to? So for cardiac disease, for example, is it actually going to the heart and not anywhere else? Long-term effects. Gene therapy is still what we consider a new approach. There's a lot that we do know about it, and there's still stuff that we're learning as just a scientific field, right? And so it's important to look at the long-term effects of gene therapy, and we'll talk a lot more about this in a little bit. And then, one thing that can be unique about gene therapy clinical trials is, as I mentioned before, typically you're comparing an investigational drug to something that may already exist on the market, right? Another approved medication. Many times in gene therapy trials, those are being tested in disease states that don't have anything available, or they're rare diseases in which there is no treatment option. And so we consider those diseases high unmet needs. And so, it's just a different consideration when the FDA may be thinking about approvals, that there is nothing else on the market to treat that specific disease or condition. So what makes gene therapy clinical trials different? Many times, the phase 1 and phase 2 studies are often combined into a single study with gene therapy clinical trials. So you may hear something like, for example, the Lexeo trial. Heroic PKP2 is a phase 1/2 clinical trial. The reason for that is, given that a lot of times we're researching gene therapy in diseases that have high unmet need, this is a way to make the drug development process a little bit more efficient, shorten the time in terms of phases of development. And so you're looking at both safety and efficacy in these types of studies. Every clinical trial, gene therapy not, has something called the screening process. However, with clinical trials, gene therapy clinical trials in particular, a part of the screening process may include something called testing for pre-existing antibodies. So this is a bit of a complicated topic, but I'll try to simplify it as much as possible. So to deliver your drug, the gene in gene therapy, you need something called a delivery vehicle for it to get to where it needs to go. Many times, that delivery vehicle is something called a viral vector. So if you were to use the example of a car, with the car analogy, the old passengers are taken out, so the virus is taken out of a viral vector, and the new passengers are put in, which is the gene that you want delivered. So you're essentially using this viral vector as a delivery vehicle to then take it to the cells that need to go in the body, the target cells. There is a possibility, though, that you have been exposed to this viral vector, just, whether you had the flu, some sort of infection in the past, previously, and that your body has developed fighting mechanisms or antibodies against that viral vector. And so there is a chance that if you have a high level of antibodies already, that if we try to introduce the same or similar type of viral vector, that your body may attack it right away. It fought it off once in the form of an infection or a flu or a viral infection. And so it kind of knows to attack the viral vector again, even though there's no virus inside. And what that exposes in terms of risk is that the gene may never actually be able to make it to where it needs to go because your body is attacking that viral vector. And so, it's just a part of the screening process with many gene therapy clinical trials. All clinical trials have predetermined safety monitoring requirements and assessments, but I would say this is kind of kicked up a notch with gene therapy clinical trials because we're still learning about them. And so what that may look like is just more rigorous requirements, more frequent visits, more assessments to really make sure that we are assessing safety properly. And then finally, long-term follow-up periods. So all gene therapy trials, after you have the initial phase or the initial part of that trial, has something called a long-term follow-up. And this is really to continue to understand the longer term effects in terms of safety, efficacy, and durability of the gene therapy. Our hope is that gene therapy is a one-time treatment, but we don't actually know that until we are evaluating it in clinical trials and in humans. And so that's why the long-term periods are so important. So here's a schematic or an example of what a phase 1/2 gene therapy trial may look like. This is not specific to Lexeo. This is not specific to any clinical trial. Like I said, all clinical trials are different. But what this shows you is just on a conceptual level, some of the stuff that we've been talking about, so you can visualize where it kind of fits into the gene therapy clinical trial process for a phase 1/2. So starting from the green bar on the left, the screening period, this is where there are some assessments or procedures done to see if an interested individual is actually a good candidate for the study. They meet all the inclusion-exclusion criteria. This is also where you may have antibody screening. There's also a very important step in the screening period where you are given something called an informed consent form or an ICF. The ICF outlines everything that you may want to know about the study, the number of visits, any potential risks, expectations, your rights as a participant. And so it's very important to fully understand the ICF and consent to that before you decide to participate in a clinical trial. Nothing happens before that. This red dot, I will consider day 1, which is the day that someone receives the gene therapy administration. It is possible, may or may not, but it is possible that you would be asked to stay at the hospital where you received the gene therapy, before or immediately after. You may also be asked to stay near the hospital for the next couple of weeks or month, after immediately receiving the gene therapy. And again, this is to really ensure that participants are being properly monitored and frequently in the beginning stages of receiving the gene therapy for the first time. After that, there are sort of these predetermined or specified site visits throughout a year or 2 years, however long the initial part of the study is. Not to say that there's, 5 or 6, there is absolutely no recipe for a gene therapy trial. But this is just to kind of show you that there are regular checkpoints or site visits throughout the study to bring you in to assess safety and efficacy. And then after that initial part of the clinical trial is over, participants are asked to be a part of a long-term follow-up period. The number of years of how long this follow-up period may be is really based on things like FDA guidance, conversations between the FDA and the company, and even learnings from past gene therapy trials. So right now, the FDA guidance is a minimum of 5 years after someone receives a dose, and it could be longer than that, depending on the patient population and the disease. Two things that I wanted to point out here that are important at the bottom: know your rights. Participation in clinical trials is voluntary, and in most cases, if not all, participants can withdraw at any time for any reason. And are you supported, right? So many times the sponsor or the company that's conducting the study has ways to support participants, whether that is helping out and arranging travel if you have to travel to a study site, covering meals, hotel stay, things like that. And so it's just important to understand what these individual trials are able to offer. Other considerations for a gene therapy clinical trial. I would, want to acknowledge that participating in a gene therapy clinical trial is a big commitment. And we're thankful to all the volunteers who choose to participate in clinical trials, because without them, we really wouldn't be able to answer some really important questions and have the possibility of bringing a potential treatment onto the market that could help so many people down the line. But it is a big commitment. Many times you may have to travel to a study site because the study site itself for the trial is not in your town or your state even, which means, time away from your family, time away from work, school, whatever that may be. And so it's just important to take these factors into account and when determining if a gene therapy trial is something that you want to pursue. Cost associated with participation. So the investigational gene therapy that one receives is at no financial cost, but again, it's important to understand, are there any potential other costs that you could come across being a part of the study, and what support is available? Is the study sponsor covering your travel? Are they arranging it for you? Are they covering your stay for you and maybe, someone to come with you so you're not alone? So just really important to ask those questions. Follow-up requirements. As I mentioned, there is a long-term follow-up period with gene therapy clinical trials. So what does that entail? Is that visiting once a year, twice a year? Is it a virtual visit? Is it an in-person visit? Just understanding what those may look like is important. And then finally, expectations. In my opinion, gene therapy is just really exciting science. There's a lot of potential and hope around it, but it's not a guarantee. The gene therapy may not work, or it may not work for everyone. And so it's just important to acknowledge that and temper expectations when participating in these clinical trials. I won't go into all of the questions here. I think all I wanted to share was that there is an extensive list of questions that's available on the GCAC website that really digs into the types of questions you can ask to understand what the commitment looks like for a particular gene therapy trial. And so if you take a look at that list of questions, it's available for you as a reference on the GCAC website. I think the most important thing here is to not hesitate and ask as many questions as you need to really understand the risks, benefits, and overall process of participating in a gene therapy clinical trial. Having conversations with the study doctor at a particular study site, your cardiology team, your primary care doctor, having conversations with your family, because it is, very important before making a decision. Okay, so where can you find information on clinical trials, including gene therapy clinical trials for cardiomyopathy? So the best first stop is www.clinicaltrials.gov. And the reason I say it's a good first stop is because all clinical trials in the US, or that are being conducted in the US, are a part of this database on clinicaltrials.gov. And so you can really see all the options that are available to someone for a particular disease area or condition. So how can you find a clinical trial that might be right for you? I'll walk you through a couple different ways that you can search for clinical trials on the website. So when you do go to clinicaltrials.gov, you will see this search field that is on my slide to the left. And there's a couple different ways that you might be able to find a clinical trial that you're interested in learning more about. So in the box that's labeled condition or disease, you can type in something like cardiomyopathy, although you'll probably get a big hit because there's a lot of trials that are looking at cardiomyopathy. So you can drill down a little bit deeper, and you can be more specific in your search and search for arrhythmogenic cardiomyopathy, ACM, ARVC, hypertrophic, HCM, dilated DCM, etc. Or if you know the trial's NCT number, that's another way that you can be taken directly to the clinical trial page of that trial itself. An NCT number, all clinical trials on clinicaltrials.gov have an NCT number. It's almost like a unique barcode. I will share the Lexeo one in a little bit. But that's one way that you can be taken to a specific clinical trial that you might be interested in learning more about. There are other ways that you can search. So in the other terms box, you can type in, the company name like Lexeo. You can type in the drug name if you know it, to kind of get you to what you're looking for. There is a search for location, although it is recommended to leave that field blank initially. The reason for that being, like I mentioned, there is a high possibility that a gene therapy clinical trial may not be in your state or your town. And so you don't want to kind of omit or exclude those options just to kind of see all that is out there in the very beginning. And so, of course, location could be a factor on whether you enroll in a clinical trial, but when you're searching for all the options, it's probably better to leave that one blank for that reason. Okay. So I'm just going to pivot a little bit here and talk about Lexeo's gene therapy program for arrhythmogenic cardiomyopathy due to the PKP2 mutation. And what I'll try to do is kind of tie in some of the concepts that we have talked about today into real life examples when we're talking about the Lexeo study. So before we get into that, just a very simplistic and high level overview of what is gene therapy. This is definitely not comprehensive. And I will say that there are some really great tools and information on the GCAC website that dig into this a lot more. There's also a webinar that was previously done through the DCM Foundation that really talks about what gene therapy is and how it's intended to work. So if you're interested in that, definitely take a look. But I wanted to provide just a high level overview of gene therapy so that it can help you understand how the Lexeo gene therapy program is intended to work. So the goal of gene therapy is to treat genetic diseases by focusing on the root cause, which, like I said, is many times a gene that is not working properly. Because a gene is not working properly, it's not making the materials like proteins that are needed for the body to function like it should. So the goal or the intention of gene therapy is to deliver the gene that is working properly, so one that doesn't have a mutation. We call it a functional gene or a working gene many times, and inject it into the body and deliver it to the cells to then be able to make the protein that you need and help the body work like it should. One important thing to point out is that for gene therapy to be effective, of course, the gene must reach the cells that need it. These are known as the target cells. And then, again, once it actually reaches those target cells, it can make the proteins and help treat or combat the disease. The way that genes are delivered, or one way, like I've mentioned, is a vector. I like to call them delivery vehicles. Again, I use the analogy of a car. So here, once again, the old passenger, which is, a virus with viral vectors, is taken out. The new passenger is put in, which is the gene that you want delivered, the gene that's working properly. and then introduce it to the body in the hopes that it will go to the target cell, in the case of genetic cardiomyopathies, the heart. So let's talk a little bit about the Lexeo gene therapy program. So Lexeo has an investigational gene therapy called LX2020, LX for Lexeo, that's designed to help address arrhythmogenic cardiomyopathy due to mutations in the PKP2 gene. So this may be familiar with a lot of you at least the top of this image. In individuals who have arrhythmogenic cardiomyopathy due to a mutation in the PKP2 gene, essentially the PKP2 gene is just not working properly. Because it's not working properly, it's not creating PKP2 protein. And we know that PKP2 protein is important in terms of the structure of the heart and how the cardiac cells communicate with each other. So loss or not having enough of the PKP2 protein results in a disturbance of the heart muscle, which can affect the electrical system of the heart and cause things like irregular heartbeats or rhythm, also known as arrhythmias, and affect the heart structure. So with investigational gene therapy, LX2020, the goal is to take a properly working PKP2 gene, so one that doesn't have a mutation, put it inside the delivery vehicle or vector. The one that we are using is called AAV-RH10. Introduce it to the body. And the goal is for the vector with the gene to then be delivered to heart cells, where it can then create PKP2 protein and help improve some of the clinical outcomes for those individuals who have arrhythmogenic cardiomyopathy due to the PKP2 mutation. Very important to point out that LX2020 is not an approved therapy. It's an investigational gene therapy, and we are currently investigating it in a phase 1/2 clinical trial. Before we talk about the trial itself, I wanted to show you this slide because it ties back into the concept that I was talking about before, that investigational gene therapies, just like other investigational drugs, don't jump right into being tested in humans. They're first tested in the lab, they're tested in animals, and the Lexeo program is no different. We did studies in labs, we collected information in animals, and we presented that to the FDA. The FDA checked that data to confirm that there weren't any major concerns with safety and that there was some evidence of efficacy. And you can see that on the green box there, improvements in cardiac function, survival, decreasing fibrosis, things like that, that justify it to move into being tested in human clinical trials. Of course, mice and monkeys are not humans. So this is definitely not an extrapolation or an assumption that you would see the same thing in humans. This is why we're testing it in humans now. But just wanted to kind of bring back that concept that there are these steps in place before a human clinical trial starts. So if you were interested in learning more about Lexeo's clinical trial, how would you use www.clinicaltrials.gov to find out more information? So there's a couple different ways that you can do that. Pulling from our example in the different fields that I was talking about earlier. So once you go on the website, there's a couple different ways you can find our study. The first top left-hand corner, you can search by the drug name, LX2020. The bottom left, you can search by the company, Lexeo. Top right, you can check by the condition or the disease. So like I said, ARVC, ACM, whatever acronym you use, or arrhythmogenic cardiomyopathy if you want to spell it out. And then finally, bottom right, this is the heroic PKP2 Lexeo's clinical trials NCT number, which is that unique barcode that every clinical trial has. I will flash up the NCT number for this study again in a little bit, but all of these different search and keywords will bring you to this, which is the clinical trial page for the Lexeo clinical trial, gene therapy clinical trial. So there you see, under interventions, LX2020, and you see that we're actively recruiting for this study. All right, so a little bit about the heroic PKP2 study. So again, this is Lexeo's phase 1/2 clinical trial. So this is a combined phase study that is testing investigational gene therapy LX 2020 in patients who have arrhythmogenic cardiomyopathy due to a mutation in the PKP2 gene. So just a couple of key features that I wanted to point out. This is a 52-week long trial, so a year, followed by a long-term follow-up period. So 5 years total from when someone receives the dose. That 5-year number shouldn't be a surprise anymore, just based on what I described before. The delivery of the gene therapy is a one-time IV injection. And the vector or the delivery vehicle that we're using, like I mentioned, is AAV-RH10. Why AAV-RH10? We have animal studies that we've conducted, and this information is publicly available. We've done some posters on this. It's also on our website. The animal studies have demonstrated that AAV-RH10 has an attraction for heart cells. And so, when injected in the body, the hope and the goal is that the vector AAV-RH10 will take the PKP2 gene to the heart cells. Again, these are things that we are confirming and investigating in the clinical trial itself to confirm. Some information about eligibility. Participants can be between the ages of 18 and 65, have a confirmed diagnosis of arrhythmogenic cardiomyopathy, and due to a documented PKP2 mutation. They also have an existing ICD that is MRI compatible. There's a lot more information around eligibility, the specifics, what does it mean to have a diagnosis, what's that criteria based off. So if you're interested in learning more and kind of digging a little bit deeper into the eligibility to see if you might be a fit for this trial, I will share some information on the next slide that will point you in the right direction to having some of those questions answered. So on the right-hand side of this slide, again, it's a 52-week initial part of the study with the follow-up period after. We are testing 2 different doses of the gene therapy to see which one is just showing a more benefit potentially. But I think the one important thing to point out here is that if you're eligible for this study and you do enroll, that you will receive the LX 2020 investigational gene therapy. So there is no placebo arm or sham, I guess some people call it. As you may expect for a phase 1/2 study, primary endpoint that we're looking at is for safety, but also looking at those signals of efficacy that you do in a phase 2 type of study. Again, this is combined, so we're looking at both. And what that is the gene actually being delivered to the cardiac cells? How much protein is being made because of that? And are there any signs or symptoms that point to some benefit to the heart? So if you are interested in learning more about heroic PKP2, there's a couple of different ways that you can do so. The first is visiting, like I said, clinicaltrials.gov. This is our unique NCT number that you can type in and find out more information about the trial itself, study sites, things like that, contact information. You can also search by the different terms that I showed before, Lexeo, LX 2020, arrhythmogenic cardiomyopathy, but the NCT number is the most direct way. If you are, really interested in learning about the study itself and what it entails, site visits, assessments, really the eligibility, you can also email us at clinicaltrials@lexeotx.com. Again, clinicaltrials@lexeotx.com. That'll put you in contact with someone at Lexeo who can then connect you with an active study site and a study doctor who can really have these more detailed conversations with you around the eligibility and what it may entail to be a part of the study. Okay, and then just a final plug for the GCAC website. Really encourage you guys to take a look at this. The DCM Foundation, in collaboration with companies, including Lexeo, have really collaborated here to try to provide as much information on things like genetics. What is a gene? What is a mutation? Gene therapy basics. So what is gene therapy? What is a vector? Things that we probably weren't able to dig into too much today. And then everything that I've presented minus the Lexeo stuff under the clinical trial section that you can refer back to or have as reference. And with that, I think that is my final slide. Before we pass it on to Q&A, just wanted to say thank you for your attention and interest in learning about gene therapy clinical trials. Without participation from the community, we would not be able to answer really important questions around gene therapy and the prospects and hope that surrounds it. So thank you. And with that, I will pass it over to you, Jayanne.
Awesome. Nilomi, thank you so much. It's really fascinating science, and it's really exciting to know where we are kind of at the cusp of all of this. So a couple of quick things here. Justine, if you can share in our deck again. Well, I think it's just the Q&A slide, actually, briefly. We're taking questions now, and I think Sandy has already been promoted.
Yes, sorry.
But if you want to, that's okay. If you want to introduce her, that's fine.
Jayanne, sorry to interrupt. Jayanne, I have the deck up. Can you see it?
I can. Yep.
Okay. I just wanted to make.
Yes. So thank you, Jayanne. So I do have, for the Q&A portion, I do have Lexeo's Chief Development Officer, Sandy, with me. She and I will kind of tag team some of the questions, do the best we can to answer them for you. There may be questions that we just don't know the answer to yet, or maybe we can't provide as much detail. But if you are, if there's something we didn't answer or you want to follow up, that clinicaltrials@LexeoTX email would be the best to follow up with. But yeah, so I think we have Sandy here. I see her. So we can
That's great.
dig into some of the questions.
Yeah, thank you for joining us as well, Sandy. We're thrilled you could be with us. So let's just dive into a few questions that have come in. A couple of people wanted to understand if you could talk just briefly again about if there's a placebo group, and do these gene therapy clinical trials randomize participants, and that's not really a thing with gene therapy clinical trials. If you can just talk a little bit more about that for folks who have questions.
Yeah, so for the Lexeo clinical trial, there is no placebo. So like I mentioned, everyone will receive a dose, if they're eligible and participate, and will receive a dose of the gene therapy. Can't speak to other trials, of course, just kind of, to the Lexeo one. And this is something that's being, talked about actively by the FDA as well, is what does that next phase of development look like? And Sandy, please feel free to chime in. When you're comparing in a phase 3, should it be against natural history? Should it be, a single arm? Should it be, we think, it's said that randomized controlled trial, so with a placebo, is like the gold standard, but there's a lot of ethical considerations around that, right? Enrolling someone into a gene therapy clinical trial who may receive other medication like immunosuppression. Also, if this is a fast or a debilitative disease, is this something that we want patients to have to undergo? And so these are all the kinds of questions that are being discussed. And I think these are, some individual questions and conversations that companies are having with the FDA.
Excellent. Thank you. That's great. People have questions about, do you have to stop taking your current medication, or are there any known medications that would interfere with gene therapy? I mean, of course, every gene therapy is different.
That's probably something that needs to be discussed with their cardiology team and the study doctor. Not every medication will have to be discontinued, but there might be some specific ones. And I don't have the names right off the top of my head. Sandy, I don't know if you want to chime in with more information there.
Yeah, I just chime in that in most cases, hopefully you can hear me okay. I seem to have problems with my video at the moment.
We can hear you. It's fine. Yeah,
okay.
Yeah, that's fine.
All right. Now, for the most part, I think, and especially for our program, participants would generally be continuing, the medications that they're on, and most of these therapies are being evaluated on top of standard of care. So it really does come down to a discussion with between the participant or potential participant and their physician in terms of what would be considered their stable regimen. We do ask that patients come into studies on, whatever they're taking, that they be on a stable dose, and a stable regimen is anticipated to continue on that. But, in terms of there really isn't specific requirements in terms of stopping or discontinuing anything else.
Okay. All right. Great. Thank you. Good to know. So I think this is the hope, but one person said, great presentation. Thank you. Do you know if having gene therapy would then eliminate the person from having a future heart transplant? And of course, that is the goal, the desire, but if you can speak at all to that.
I mean, I think we're in the beginning phases of looking at the gene therapy and how it works in the human body. So not something that we can probably answer at this time. And how exactly, what the benefit or the clinical outcomes of the study or the investigational gene therapy will be. But, I think the hope is doing as much as we can to address the underlying cause.
Okay, thank you. This is an interesting question that I have also had. I don't know if either of you can answer. This woman has the PKP2 gene, but had a heart transplant 2 years ago. Is there any chance the gene can be passed on to the new heart? Do we know the answer to that question? Sandy, would that be something you might be comfortable answering?
If they received a heart transplant, in general, the genetics are those of the transplant organ donor, not that of the recipient.
Okay, incredible. And my understanding was that heart cells don't turn over that quickly. So therefore,
that's correct.
Okay, so therefore it's pretty much you end up with whatever the genetic makeup was of the donor for the heart. Okay, incredible. Thank you. So let's see, this is an interesting question. How do you assess if the gene has reached the heart muscles? Is this done with a biopsy or an echo or something else?
Go ahead, Cindy.
Yeah, so we do, within our trial, and I do believe in several other trials, it is of interest to get tissue from the target organ of interest. And so in the case of cardiomyopathy, there is often a requirement in these trials for the participants to undergo a cardiac biopsy, generally one at baseline and one at some time point following the administration of the gene therapy. And that's really the best way to know if the vector has reached the target organ and target cells, and then actually understanding if it, not only that it gets into the cells, but does it actually start producing the protein that's of interest.
Okay, excellent. Interesting question. Not sure if we can answer it. What is the best case scenario of when the gene therapy for PKP2 might be commercially available and FDA approved? I understand this could be a year's estimate, decades, whatever. Is it expected to be able to return? Would someone be expected to be able to return to their full regimen of professional sports or bodybuilding, etc., and be fully active if the treatment were successful? I know this is all a bunch of what ifs, but
Yeah, I mean, I'll take at least the first part of that question. Hard to say exactly a time period. We do the combined phases, as a lot of the other companies are doing, to try to shorten that duration. But as you saw in my presentation, these trials, whatever phase it may be, could take several months to years. And then there's in between period where you're sharing that data with the FDA, understanding what a phase 3 or a larger study may look like, and then the time that it takes to then enroll that trial and assess the data there. And so, years is probably the answer, but I, don't have anything in particular that I could share about that.
Yeah, I agree with that.
Okay. Excellent. And we have just a few more questions and a few more important slides. So if everybody can just hang on, we may just run a few minutes after 7, if that's all right. I'm not sure if we can answer this question. What percentage of the population is estimated to have antibodies to the AAV vector? And does it depend on the vector? I mean, there's different AAV therapies are used, right, depending on the trial.
Yeah. Do you want me to comment on that? So
Yeah.
it does depend on the, which viral vector is being used. And it also depends, sometimes it depends on geography. It also depends on the age group of the patients. Of course, generally older patients have a longer period of time in which to have been previously exposed to a virus that can, cause the body to have a prior immune reaction to a prior infection or virus that could potentially leave that patient with antibodies that can preclude the patient from being eligible for the study. So I would say it ranges. It can be anywhere, there are different studies that could show a range anywhere from 20 to over 50% may have underlying antibodies. Nilomi, I don't know if there's anything else you'd like to add to that.
No, I think it just, again, underscores importance and why you may have antibody levels as a part of the screening process and eligibility for some of these trials.
And I know longer term, they are looking at ways to kind of work around that. And so they are doing studies on how to deal with folks who may have been exposed to those viruses, but that's longer term. Let's see. Let me just see what else. And what is involved with immunosuppression if you were to take this gene therapy clinical trial, if you were trying this?
Sandy, do you want to take that one?
Yeah. So with most of the clinical trials, they are utilizing some form of immunosuppression. Often it includes something like corticosteroids or prednisone, but it can include other stronger immunosuppressants. And it really depends on, it really varies, I think, each study, each program, depending on the indication, the vector that's being used, how much of the vector is being administered can sometimes determine how much immunosuppression is going to be used. And I would say typically it's for several months following the administration, following receiving the dose of the gene therapy. Typically, it's not for long term. It's usually just for the first few months.
Okay, excellent. Oh, that's encouraging. And we have a few more questions that we didn't have time for, but we certainly will follow up with email to those individuals with their questions. People are asking about other genes, of course, and what therapies are coming for those. And so this is such an exciting time right now for gene therapy in cardiomyopathy, especially. So I will be answering and responding to each individual, but just know that there are therapies coming, and depending on the gene, some are sooner, on the horizon than others. But thank you both so much for your time, for the presentation. If anyone has questions that you didn't ask or didn't feel comfortable asking, feel free to email me directly. And my email address is jrock, JROCK, @dcmfoundation.org. And please do visit our websites for more information. Nilomi, Sandy, thank you so much for being here tonight. We just greatly appreciate your partnership and great presentation. And obviously we had, a lot of interest and great questions. We had 321 people registered for this webinar at last count. So this is really a very hot topic. And I think the interest in this is just going to grow like a tsunami here as we go forward and in such an exciting time. So thank you again. I really appreciate you being with us tonight. And Justine, if you can share in the deck, we just have a few other things to note, and actually a few other tidbits, which I will also send via email. So first of all, we do now have a number of gene-specific Facebook groups that we would like to promote. If you go to Facebook and you type in the search bar any of these that are here on your screen, those are Facebook groups that are specific to those genes. If you have that gene, please go ahead and join those groups. And that's where we are going to be sharing information and keeping people updated about upcoming research and information about clinical trials and so forth as that becomes available down the line. Obviously, some of these genes are being researched earlier than others. It just depends on a number of factors. But so if you see the list here, there are now groups for BAG3, DSP, the LAMP2 gene, Lamin A, MYBPC3, MYH7, PKP2, the Titin gene, TNNT2, TPM1. And if you don't see your gene listed and you're interested in starting a group, please email us at info@dcmfoundation.org. And those emails come to me, and then we can set up a time to talk about what it would take to start up a group. So we're very excited. We'd love to have another 50 groups in the next couple of years so that we have like almost all the genes. Eventually, we would love all the genes to have a specific group where people with that similar gene can join, share information about their experiences, and learn about upcoming research studies and trials that are going on for their gene. So really exciting time, super incredible stuff happening, super partnerships between researchers and industry folks and patient groups. And it's just a great time to be like looking forward and having a lot of hope. So next slide, please. So again, for more information, oh, I think we're stuck on a slide. Well, for more information, please go to geneticcardiomyopathy.org, and you can always type in clinical trials in the search bar, and you can find a lot of information there. We've worked diligently to have very clear and concise information. Justine, are you able to forward the slide deck? I just want to give a huge...
Yes, I am. Where I'm looking for is the information one.
Oh, that's
so
amazing.
Do you see this at all? Okay.
That's okay. We're on the gene specific one. I just wanted to give a shout out to all of our generous sponsors and all of our partner groups. I think you'd have to reshare in the deck.
That's what I'm thinking. Let me just let me see if
maybe just go to the sponsors page then, because it's really the most important.
Let me do it near. Sorry about that, guys. It's a little wonky today. Can you see that?
That's okay. Oh, it's not switching, but that's all right. We also, I want to thank all of our great partners. We have a number of cardiomyopathy. There we go. Yay.
There we go. Okay.
So these are all of our partner groups, and these are different patient organizations that we recommend that you check out. Not sure if you can go to the slide before, but that's the sponsor slide, if it would work there. One forward. Right there. So these are all of our amazing sponsors. I love working with every single one of these companies. These are amazing folks doing great work, and we just so greatly appreciate their support. And without their support, we wouldn't bring you this incredible information. So, and if we just want to go to the last slide, we invite you to register for our next webinar, which is already up on our website. It's about insights and hope when living with a device. For anyone who's contemplating getting a device, if you have a heart device, an ICD, an SICD, pacemaker, any of those things, please join us. Doug Raychak, who's a rock star about devices, he's going to share with us a lot of information about that on Wednesday, July 17th. So we hope you can join us then. Thank you for joining us. Thank you for your questions. An email tomorrow will be sent out with more information and the webinar recording. And I don't know where all you are tonight, but I hope you're staying cool. Wherever you are, stay cool and stay healthy. Thank you all, and thank you to Justine, and thank you to Lexeo and Nilomi and Sandy again. Take care and have a great night.